So, thank you very much. My name is Paresh Vyas. I'm a haematologist. So I treat mainly blood cancers in the Churchill Hospital here, but I also have an appointment with the MRC Molecular Haematology Unit in the Weatherall Institute, and I work very closely with my colleagues in the Biomedical Research Centre here. So today I've slightly rethought this talk because of the audience. So I was going to tell you something about tracking cancer mutations through treatment and how that's important in terms of precision medicine. But it struck me actually in conversations that we had with my colleagues around the country about some of the advances we've made in delivering stratified trials and what it's taken to do that and how that may be really of value for the community, the cancer community at large, but also other communities. And so you all know that cancer is mainly a disease of older individuals, that cancer is increasing in aging population, and we are understanding the genetic basis of this. And this has important implications for patient risk stratification, identifying targets, identifying less toxic therapies, and particularly in older people, this is very relevant, and defining combination therapies. And Anna Shu and others would have told you about this. But there are real issues that trials are expensive, they're slow, reimbursement is challenging, and of course reimbursement is easier the greater the impact of the drug, which means smaller patient populations, and that's a trade-off industry, and the payers have to really work through. But I think what we need to be working towards, and we've talked about this today, is the genetic characterization of all patients. And in particular, I'm going to be telling you about new national trial infrastructures, really to allow rapid recruitment and to incentivize hospitals. So now there are many structures within the NHS to try and promote research, but not all of these structures are working properly, and still recruitment is slow. And if you look internationally, the United Kingdom, despite having an integrated healthcare system, doesn't always recruit at the top of the tree. And this irritates the industry. We also need detailed characterization of patients progressing through trials, particularly with real-time PK/PD, and I'll show you an example of that, deep scientific analysis of sequential samples through therapy, and we also need to collect in the later phase studies quality of life and impact on healthcare resource utilization, and the DRC has been very thoughtful about that here in Oxford. And I've been helping both NICE and also working with the MHRA on some of these issues, and this is really important, and I don't think pharma thinks about it enough. And this, I hope, will allow us to identify responding and non-responding patients. I'll talk about that in the last bit of my talk. I'm not going to spend too much time on identifying mechanisms of response and resistance, but we can do that by deep scientific analysis. And as I said, we have to work with regulators and reimbursement agencies. So the NHS has some huge advantages. It's a single healthcare provider, high quality clinical teams, we can put genomics and stratified medicine programs, and we have trial units. But there are real problems. And the problems are really that there is a shortage of there's a shortage of research and support for these trials, and we lack network catchment areas really for the delivery of complex trials in genetically stratified cohorts. And we need large populations when we're looking for subpopulations of genetically homogeneous patients. So together with my colleagues in Birmingham, we set up a trials acceleration program, which was initially funded by philanthropy and blood cancer charities, but now has some commercial underpinnings, and also a stem cell transplant cooperative, the first in the world really to help trials in the disease I look after, which is acute myeloid leukemia. And the aim is to create capacity and confidence to deliver everything from phase 1 to phase 3, to access innovative therapy. So rather than the trials being done initially in the US or on the continent, they're done in the UK, clinicians can get experience, clinicians can get confidence with drugs, and to deliver within that through sequential sample analysis, translational research. So in the last 5 years, we've opened 12 new clinical trials in early phase, 7 in late phase. Trial setup time is short, recruitment is rapid. We deliver pharmacovigilance to industry standard, and there's been a lot of interest from pharma now. So in addition, we've embedded science in these patient sample studies, and I've been doing this really in the Weatherall Institute of Molecular Medicine, which I showed you up there. We have an MRC unit there, we have quite a lot of funding, we have a lot of infrastructure, scientific infrastructure, and it's all under one roof. And in contrast to where pharma outsources samples to lots of different vendors, you really have something within one hub. And this maximizes patient sample use tremendously, and this is really quite important. So what we've done is really provide an opportunity in early phase trials where we can really, we've realized that actually a lot of the preclinical data in cell lines or in animal models and xenografts, all of that stuff, actually you really have to start again when you start looking at drugs in patients. And you really need to really deeply analyze the genotype and function in real time. And in later phase trials, it really helps determine drug combinations, and I'll talk about that. And I've talked about real-time PK/PD, and I'll talk about resistance mechanisms. So to do all of this, we've been banking bone marrow samples because we're interested in blood diseases. We've banked about 6,000 viable cells from bone marrows over the last 5 years. We have a very good understanding of our disease. We understand the downstream assays really well. And we built an academic lab environment, and we think that this may now transition out into a sort of semi-commercial environment. So I'm just going to tell you really about 2 case studies that really exemplify this network. And I'll just tell you a few words about acute myeloid leukemia. So it's the most aggressive common leukemia in adults. It's still quite rare, 3,000 cases a year in the UK, but the survival is pretty poor. 70% of patients will die within 6 months of diagnosis. So it's a pretty aggressive disease. Most patients are older, and we really haven't improved the outcome of these patients in the last 3 decades. So some 7 years ago, we started a collaboration with Stanford University on a therapeutic monoclonal antibody targeting cell surface CD47. And the scientific basis for this collaboration I'll just set out in this slide. So phagocytic cells really eat or remove unwanted cells. And here I'm just showing you a cancer cell in blue, a phagocyte in grey, and the eat me signal results in phagocytosis. But all cancer cells, including ML cells, express don't eat me signals, and there are only 2 don't eat me signals, CD47 and CD200. CD47 is a major don't eat me signal. So in collaboration with Stanford and the Weissman lab, we produced a don't eat me blocking antibody, anti-CD47. And what that does is it really encourages phagocytosis of cancer cells. And we went from those beginnings with MRC funding, with California Institute of Regenerative Medicine funding, and then more latterly with investor funding in something like 6 years from an antibody that recognized in urine CD47, so a humanized antibody to full-scale production through all the regulatory preclinical tox data into a phase 1 study that started about 20 months ago now. And this is first-in-class, first-in-man study. We have help from the BRC here. The total spend is only about £11 million. And phase 1 study is wrapping up in about 2 or 3 months' time. And the phase 2 studies are planned to start later this autumn. And we'll be presenting this data later this year, the efficacy data. But suffice it to say, the signals are really interesting. So this was a collaboration with Weissman and Ravi Majetti, who are pictured up there. So this is an example of taking observations from basic science through to a phase 2 in pretty good order in a cost-effective way, and using the infrastructure that we have in Oxford to help us do this. The second story I want to tell you is about stratified medicine, really. So there are mutations in a gene called isocitrate dehydrogenase. It's a Krebs cycle enzyme. I don't have time to talk about the biology of this, but essentially 8% of AML patients have this mutation in IDH1, and 15% of patients have a mutation in IDH2. And I'm going to concentrate on the IDH2 mutant patients today. So we can identify this group of patients genetically, and in collaboration with a company in Boston called Agios, they discovered a small molecule inhibitor, AG221, and what it does really is that it binds the mutant enzyme selectively. It's about 10,000-fold more selective for the mutant enzyme than wild-type enzyme. It's an allosteric inhibitor, and here I show you AG221 between the dimer of the mutant enzyme, and it just holds the mutant enzyme open, preventing the enzyme from working. And this is xenograft data from a paper that was published with us this year, and essentially you can see that this is the survival curve with vehicle in blue, and then increasing doses of drug to survival in animals that were treated at the top dose. But in this xenograft study, of course, which was highly selective in terms of patient samples, all the mice survived. And the remarkable thing that this drug does is it causes the cancer cells not to die and has a cytotoxic effect, but it causes the cancer cells to differentiate. And I'm sorry, I can't do so you can see here, these are immature cells, you have to take my word for it. These are mature cells. So it causes cancer cells, if you will, to grow up. And this is the phase 1 study, phase 1, phase 2 study that has just been published, and it involved Memorial St. Catherine, France, and centres mainly in the United States, but the lab work behind it was done in Oxford. And so this is heavily pretreated patients, older patients, 67 years old, and the median response was 40%. And I'll spare you the clinical details, and these are the survival curves. So here you can see patients who are not responders, and 60% who didn't respond, and these are the patients who responded. So the data from this clinical trial tells us two things. Firstly, it's very impressive single-agent response. This is an oral tablet. This can be taken by anybody, and the side effects are absolutely minimal. This really targets the mutant enzyme only. But unlike the preclinical data, not all patients respond. And unlike the preclinical data, the responses were not curative. Why was that? Well, we showed, and I don't have time to go through the data, that the mechanism of action of this drug really involves the suppression of an metabolite, 2-hydroxyglutyrate, and when you take that metabolite away, it releases epigenetic processes to restore multilineage, i.e. many different types of blood cells were made from arrested stem and progenitor cells. We also showed that the primary mechanism of failure, i.e. the 60% who didn't respond, it was the nature and number of coexisting mutations, and in those patients who had a response and then lost the response, what we call secondary resistance, the basis of the resistance was not second site mutations in the drug, i.e. In the mutant enzyme, I mean, so i.e. that it wasn't that there was another mutation in the IDH molecule that now made the IDH molecule resistant to the drug. So essentially, you shouldn't, in those patients who are relapsing, other companies' compounds are unlikely to work. And essentially what happens is that the drug selects therapy-resistant clones. Now I'm just going to show you that. We did whole exome sequencing, and in some cases whole genome sequencing. Then we did single-cell sequencing to really work out the order of mutation acquisition. So this is a cell, the initial wild-type cell. This is a mutation in a gene called ASXL1. I won't go through all the gene names, but essentially each letter is a mutation in a different gene. And you can see the hierarchy of mutations, and then at the end, the clone splits. And this was the major clone that was operative in the patient pre-treatment. And with treatment, this clone really shrunk down, and this clone differentiated and gave rise to mature blood cells. So these mature blood cells gave rise to, a functioning blood system were mutant. They were all leukemic. And with relapse, what happened is that this clone hadn't disappeared completely, and there were additional mutations that were identified. And in each patient, we could identify a whole series of additional events that occurred. And we've now looked at about 50 to 60 patients in this way, and we're beginning to understand the pathways that give rise to secondary resistance. So this has allowed a new drug to be born. It's called anacetinib, and Celgene owned this drug. We went from first patient, first visit to an NDA application, I apologize for the typo here, in 26 months. This is really rapid. And this drug will, I suspect, get its approval, fast-track approval from the FDA later in August. We provided the data for the mechanism of action and data on molecular stratification of responses, duration of response, and relapse. And we now have some phase 2 randomized trials coming up to really look at relapsed patients. So this has been a real success, I think, for Oxford. And I think the next steps in learning from this, so we can leverage academic NHS and UK life sciences infrastructure, but we have to really rethink our current model of phase 1 to phase 3. And certainly in cancer, if we use our early phase trials properly, we can get to licensing quickly. I think the current CRO models are very expensive and inefficient, and pharma's understanding that, and we need new structures to deliver faster, clinically relevant, biologically stratified trials from large populations to regulatory standard with real-time deep analysis to make data-driven protocol changes. And I think this faster development will allow for faster combination trials. And so I think we need the combination of the academic sector with key opinion leaders, the NHS and industrial partnerships to drive this. And this really embraces the best of what is experimental medicine and patient-centered care. So I'd just like to thank, there were a large number of people involved. I'm just acknowledging a very senior clinical fellow in our lab, Lynn Queck, in Birmingham, Charles Craddock, who's led the whole therapy acceleration program. We collaborated internationally and with companies, but I'd particularly like to thank Oxford University, the RDN, the Radcliffe Department of Medicine, and the WIN for allowing all of this.